Okay, good morning. Today we're going to talk a little bit about mammalian gene expression of clone genes in prokaryotic as well as eukaryotic cells. Now there are some issues that are important for that, and we'll go over those, at least some of those, a few at a time. So in terms of why we want to put mammalian genes into mammalian cells or prokaryotic cells, sometimes the mammalian proteins just don't work in prokaryotic cells for some reasons I'll explain in a second. And the other issue is when we're going to be using clone genes, mammalian clone genes, we generally use cDNA inserts rather than the full-length gene. And again, there's some serious explanations for why that is actually true. Okay, so one issue that becomes important when you're trying to express mammalian genes, especially in prokaryotic cells, is that of codon usage. Now, everybody's familiar with the idea that some amino acids can be specified by more than one codon, but that does not mean that all those codons are used equally. It turns out that they are used very differently in mammalian versus E. coli cells for certain of the amino acids, and this can lead to poor translation of some eukaryotic proteins in prokaryotic cells. There are other issues as well besides that, poor translation, that prokaryotic cells don't have the same mechanisms for modifying proteins that eukaryotic cells do. Importantly among these is glycosylation, but also phosphorylation. We don't have the ER pathway in prokaryotic cells that is going to be able to carry out those reactions for us. So there are a lot of important differences in the basic cell biology between eukaryotes and prokaryotes, which come into play when you try to express a prokaryotic cell, eukaryotic gene in a prokaryotic cell. Right, so here's the idea of codon usage. Eukaryotes and prokaryotes have the same genetic code, which is to say the same codons are used to encode the same amino acids. Now there are some exceptions for organelle genes, but we're going to talk about that at a different time. Okay, but this can be a problem when we're trying to express mammalian proteins in E. coli because the codons that encode the same amino acid are not actually used at the same relatively frequency in all organisms. And this influences the levels of the tRNAs that we're going to have to bind to those codons. So the one example we'll do is arginine, but it's not the only one. This is just the most extreme example. Okay, so here's our universal genetic code. This is very familiar to us. And what we're going to be talking about is the arginine codons. Now, there's 6 arginine codons, right? All the CG somethings and the AGA, AGG all encode arginine. Okay, and it turns out that those are not used equally at all in different organisms. But most importantly, the usage actually is very different between E. coli and mammalian cells. Okay, so here I circle our arginine codons, and you can see that there are 6 of them. Okay, so here's the result of one study, and there have been many, that actually looked at the differences in arginine codon usage. Now for us, we're considering the human and E. coli comparison, where we've got our 6 arginine codons listed on the left, and then the frequency of their usage of that particular codon in human. So for example, our AGA codon is used 23% of the time in human, which is to say 23 occurrences of arginine codons are AGA. Similarly, AGG is a substantial percentage of actually used arginine codons in human. But if you look at E. coli, it's only about 1% for each of those. And the most preferred E. coli codons, which are the CGC and CGU, are used relatively more in E. coli, but not used nearly as frequently in humans. So this difference in the codon usage is reflected in the tRNA population, because when you have a codon, you need to have a tRNA that's going to recognize that and carry the arginine to the ribosome for the purpose of synthesizing the protein. So this does have bad effects on translation in E. coli of those AGA and AGG codons. Okay, so these are the two that are rarely used in E. coli, but are much more frequent in mammalian cells. So the tRNAs that recognize these are relatively less frequent in E. coli versus the ones that recognize the other form. So the effects of this is that the mRNA encoding a mammalian protein that has a lot of those troublesome codons is going to have trouble getting translated in E. coli, because the translation process will have to wait when that codon is encountered for the correct tRNA to show up. And that can lead to stalling of the ribosome, or else the ribosomes can just fall off entirely, or it can cause frame shifting, which is not a good thing. Well, there are some workarounds that people have found, which are not necessarily convenient, but will increase the expression of the mammalian proteins that are compromised by the use of these relatively rare codons. Okay, so one is they're actually bacterial cells, E. coli cells, that have been engineered to contain extra copies of the gene encoding that rare tRNA. So the rare tRNA is not as rare anymore because we're having extra copies of it there in the cell. And so these are actually commercial strains, and they're sold by various suppliers, and they simply have more copies of that tRNA, and that certainly is going to help translation. The other relatively non-preferred way to deal with this is a process that we'll talk about some other day, which is site-directed mutagenesis, that it's possible to basically look at those codons and use this procedure to change them to one of the preferred E. coli codons. Okay, so another problem with mammalian proteins is that E. coli cells do not have the enzymes to properly glycosylate, phosphorylate, etc., mammalian proteins that would occur. Now, sometimes this doesn't matter, that your serines or asparagines are not glycosylated, but sometimes it does cause the protein to be ineffective, that the protein, for its proper function, does require those modifications. Okay, so let's remember what these are. The sugars that can be added to proteins, for example, glycosylation. Okay, so what's shown here is a serine or threonine with an O-link that's been attached by means of the hydroxyl group on those amino acids. And this could be a simple sugar, or it could be a very large, complicated, multi-sugar structure. Okay, but these glycosylations, or addition of sugars to protein, is very common and characteristic of mammalian cells, especially those that go into the membranes or on the cell surface. Okay, so the other type is asparagine linked, and this shows asparagine. The green thing represents the protein sequence, and its side chain is sticking out, and it's attached to a sugar. Now, the sugar itself could be just a landing site for a very complicated sugar structure, and these are also very common in mammalian cells. And the E. coli does not have the mechanisms in place, the enzymes, to create those sugar residues, and that could have bad effects on whether the protein is going to be functional or not. And so if you're trying to create a recombinant protein for some therapeutic reason, it needs to be able to work correctly. And so this is a situation where you've got to do something about it. Okay, so here's an example. It's a Western blot, and it's used to detect the protein beta-glucocerebrosidase, which has been made in E. coli versus what's been made in a particular cell line, in which case this is called Chinese hamster oocyte, or CHO cells, as compared to E. coli. So we'll hear more about CHO cells in a sec. But the idea is that the gene was cloned on a plasmid and expressed in E. coli, and that's on the right lane. And so you can see there's a very strong band at the molecular weight position of the unmodified enzyme. Now, if we take the same enzyme and produce it in mammalian cells, we see these are all different cell lines, a molecular weight shift where the protein is considerably larger when it was produced in the mammalian cells or cell line. Now, that size difference, it turns out, is due to protein glycosylation, which the CHO cells are able to do perfectly well, but the E. coli cells are not. And it turns out that if you have a situation in E. coli, that protein does not work, doesn't correct the genetic defect of this Gaucher's disease, whereas the one that's produced in mammalian cells works very well. So it's important to produce this protein in the proper mammalian cell line in order that it will have a therapeutic benefit to the people that have that particular disease. Okay, so this says what I just said. Okay, so the take-home message here is that sometimes E. coli cells just will not do. And what we need to do is take our clone gene, which we put together in E. coli, and then transfer it to a mammalian cell for the purpose of producing the protein that we want, right? So the question is, where do people get these cells? That these are actually cell lines that are used rather than just mammalian tissue, and they're largely purchased from repositories of such cells. Okay, so HECC is one example that if people want to use a certain cell line, they will acquire it from this place, and then they'll bring it home to their lab and then propagate it further for whatever experiments they want to do. And there are thousands and thousands of cell lines, but some, of course, are more popular, more widely used than others. So this is the HECC repository. They're purchased, brought home, and then cultivated in the individual labs. Okay, so this is not a trivial situation. Mammalian cell growth is a whole lot more difficult than your usual E. coli cell growth, that you need to provide the cells with what they want. You're trying to convince them that they're not really in a culture dish, they're really in a mammalian body. So you're trying to basically mimic those conditions in a culture dish. So your temperature needs to be close to 37. It needs to have a CO2 level, which is like what it would encounter in nature. There are many times very complex growth requirements. You can't just give it a little sugar and salt and think it's going to grow. It's not. They need growth factors, sometimes complex nutrients, vitamins, and very importantly, they grow very slowly. Their generation doubling time can be several days, really, not always that long. But it's not the 20 minutes that you would get for E. coli. And then also you've got the situation where you're giving them this wonderful complex growth medium that they just love. Well, your bacteria and your fungi like that too. So preventing contamination by the organisms in the environment is a real serious consideration when growing mammalian cells. Okay, so here's a picture of a special hood that's used. It can be kept sterile inside, and its purpose is to keep contaminants out of mammalian culture. Okay, here's a picture of this has got a slide inside it, but what the cultural medium looks like. And it is a complex medium, includes serum and many, many nutrients. So in general, about mammalian cells, you can't just get some mammalian cells from the body and expect them to be able to propagate and divide in culture. So generally what has been done to get these cell lines is that they did derive from a naturally occurring cell, and they were removed from the body or from the cancer sometimes, and then grown in culture. So the primary cultures don't really thrive too much, but you get a few cells that do thrive because they've experienced genetic changes that enable them to continue to divide basically indefinitely. So those cells are considered immortal, and those are the ones that can be propagated endlessly. And those are the ones that are largely done for experiments in cell lines. Okay, so they differ from their starting cells in that they have changes that allow them to violate some of the rules that mammalian cells have. Mammalian cells don't like to grow independently in liquid. They like to have a surface to stick to. They require growth factors of many sorts, and they refuse to grow when they're in contact with other cells. So these are general properties of mammalian cells in the body, which the immortalized cell lines have had genetic changes that allow them to escape. But nonetheless, they look a lot like the cell lines, the cells that they started with. The fibroblasts, for example, they're long, skinny-looking cells from connective tissue, and the cell lines that are made from fibroblasts still look like that. So they've had genetic changes, but they still resemble their original cell type in many respects. So you can have fat cells, adipocytes, hepatocytes from liver, and many others. Now, one you may have heard of is HeLa cells, which is originally from the cervical carcinoma, cancer cells, and these were the first human cells to be successfully cultivated, and they're still very widely used. And the other famous one that we'll talk about in a sec, which we already mentioned, is the CHO, the Chinese hamster oocyte cell line, which is used for industrial purposes for making many recombinant proteins, including vaccines. Some other famous cell lines include ES, or embryonic stem cells, but also somatic stem cells. Okay, so here's a picture of some fibroblasts. They're long, skinny cells. They're not really green. They look green because they're expressing GFP from a plasmid that somebody put in it. The blue is actually the nucleus, which has been stained with the DNA-stained DAPI. So you can see that they still are elongate in the culture, which is what they look like in the connective tissue in the body. Right here are CHO cells. They're not been stained with anything, but these are really good growers that are very commonly used. I know that sounds like a ridiculous Chinese hamster over. That's sort of historical. But these are actually extremely important. It's the workhorse of the biotech industry, or so I'm told, that it's just used for producing mammalian proteins for therapeutic purposes. And these have been very much manipulated to produce different proteins at very high levels and are very commonly used. Important feature of these is that they grow in suspension. They don't need to be attached to a petri dish to grow. So they can be grown in these gigantic containers, which is what is needed to produce large amounts of these various proteins. So here's a picture of gigantic tanks that are used for the industrial scale production of recombinant proteins. Most cells can't grow like that. Some examples. TPA is tissue plasminogen activator. Things like insulin for diabetics. Human growth hormone are made in these cells. Many vaccines are produced in these cells. In fact, over 70% of industrial recombinant proteins are produced using CHO cell lines, which have been individually manipulated for production of those individual proteins in different kinds of ways. And in fact, some of the COVID-19 vaccines that are under development use these cell lines. So some of them have been manipulated. They're not their original natural self either. They've been manipulated to produce human-like glycoprotein modifications, which are apparently not identical to what the hamster does. So these are very manipulated cells even before you start getting them to produce the recombinant protein. Okay, so once we've chosen a cell line, we need to be able to get our DNA in there, which sounds like it should be difficult, but it's strikingly easy actually. It's actually much easier to get DNA into cell lines, mammalian cell lines, than into E. coli or lower eukaryotic organisms. Okay, but one more little term before we start discussing this. The word transfection is used for the process whereby you're putting DNA into a mammalian cell. Now, we think of that as transformation because that's what the term is for E. coli. But instead, in mammalian cells, transfection is used rather than transformation. And there's a couple of reasons for that, but one is that the word transformation in mammalian cells, historically, is used to describe the change when a normal cell becomes transformed to a cancer one. So basically, the word transformation was already taken in the mammalian cell culture because the process of normal becoming a cancerous one is considered transformation. But transfection is most like what we think of as transformation. All right. So in getting ready to get a recombinant protein produced in a mammalian cell line, we need to start with E. coli to get the gene of interest inserted into a plasmid vector, usually, not always, that will eventually be able to overexpress the protein of interest in the mammalian cell line. So the original experiments using E. coli will have chosen a vector that's going to be able to do that efficiently in that cell line. So once the plasmid is put together in E. coli, it needs to be introduced into the mammalian cells for the production of the desired protein. Now, once it's there, there's two kinds of experiments, really. One is what's called transient, where we're not actually selecting, and the cells will eventually lose the plasmid as they grow. But those transient experiments are used for a lot of things. But we can get stable cell lines in which the plasmid itself is incorporated into the chromosome of that cell, and that cell will express the protein permanently because it's now part of the mammalian genome. So those are basically two separable processes. Okay, so here's a plasmid vector used for transfection, and it has the CMV promoter because that's going to drive transcription of the production of the RNA in the mammalian cell, not the E. coli one. And we're using this ORF, or whatever protein coding region we have, which is in the form of a cDNA, and it's fused to GFP, and that's going to enable us to see that we've produced the protein correctly in the mammalian cell line. Now, some of the other stuff we don't need for either purpose, but some we do. The ampicillin resistance gene is there because we needed that for selection in E. coli, and the choline one is a replication origin, also for E. coli. We're not going to need the neomycin selection, well, G418 selection for mammalian cells for most of our experiments, but it is present on the vector. There's also an SV40 origin, which again we are not going to need for most experiments, but that will allow replication of the plasmid in the mammalian cell. So for this purpose, all we really need right now is the promoter, the ORF, and the GFP. Okay, so transfection is the process whereby we're getting nucleic acids into animal cells. So it's analogous to transformation, but the huge difference is that it's way, way, way more efficient. Like in E. coli, you get transformation, but it's very rare, and that's why you have to select for it. So if you add DNA to an E. coli cell, then you need only very, very small percentage of the cells actually take up the plasmid and replicate it. Whereas in mammalian cells, the frequency can be very high, 50% or more in some cases. Okay, so in this case, what we're going to do is take a molecule that produces our protein, or we think is going to produce it, an ORF driven by the CMV promoter, fused to GFP. So the way we're going to do this is one process called liposuction, which is not the only way to transfect, but is very popular. Okay, so for liposuction, the DNA or RNA is assembled into lipid particles by using a reagent called the lipofectamine reagent, and this will spontaneously encapsulate the nucleic acid inside this little particle. Now, this very much resembles the mRNA vaccines that were being given from Moderna, etc., in that what you have is your nucleic acid inside a little particle, which is going to be able to deliver it into cells. Okay, so when introduced to the solution with the mammalian cells, the lipid particles bind to the surface and are taken up by the mammalian cell. We don't need this. So here's a diagram of the transfection process. We have a nucleic acid of some sort that we want to encapsulate. It could be a plasmid, an RNA, or it could be a different type of RNA. The nucleic acid is incubated with the transfection reagent, which is lipofectamine, and it forms a complex in which the nucleic acid is on the inside and it's coated with this nice layer of lipids on the outside, which is then binds itself to the cell surface by means of its lipids, and then is taken up by the mammalian cells by a process which is believed to be endocytosis. It's certainly like endocytosis. So once the particle is inside the cell, has been taken up by it, then the nucleic acid has to escape the endosome, and then it's free to either be translated directly, if it's an RNA, in the cytoplasm, which is what would happen with the Moderna vaccine, or else the DNA would make its way to the nucleus, where it would be transcribed by the host cell RNA polymerase II, and then the RNA from that would then come back out into the cytosol for translation by cytosolic ribosomes. So we've taken over the cell's machinery for, in some cases, transcription, in other cases, just translation, for the purpose of producing the protein that we are trying to produce in these cells. So here's the process I just said, pretty much in outline form. The reagent forms complexes, which are then delivered to the cells, where they escape and then are used by the host cell machinery to produce RNA and or protein. Okay, so the first type of experiment, or most common really type of experiment, using mammalian cell lines, doesn't really aim to have the cell produce the particular protein endlessly. It just wants to see what's happening in the first few days initially of when it gets in the cells. Okay, so the important parts of the vector here again are the CMV promoter, the ORF, which is fused in frame to GFP to produce a visible change in what the cells look like as a result of expressing GFP. Now remember, if you're producing this mRNA that's where the ORF is in frame with the GFP, you'll produce this fusion protein where in this case where the N terminus is the protein of interest and the C terminus is a GFP. So we'll be able to see the location of the protein of interest because of the GFP. We'll be able to see the GFP, and we will infer that those two things are in the same place because they're part of the same protein. So this plasmid has a promoter, the fusion. It also does have a neomycin resistance, which we're not using in this kind of experiment, but it does have a ampicillin, which we used already. It does have replication origins, which again we only use in E. coli. Okay, the important thing here is that transfection is so efficient that you can basically introduce your nucleic acid to the cells and just look at them 24 to 48 hours later. And we'll be able to see with the GFP that many, or most of the cells in some cases, have actually taken up the molecule and produced the protein from it. And so this is basically for short-term experiments, which many are, which doesn't require selection in the mammalian cell at all. So here's some pictures. This is from an ad for a transfection reagent, so you can take that for whatever it's worth. But the cells that are green is because GFP has been expressed from the transfected plasmid. And so there's a bunch of these different cell lines, and this is most of them, or all of them, really produce significant amounts of GFP, some better than others, but there's very clearly been a successful transfection here because, for example, HeLa cells are not normally green. So this is the standard, typical type of experiment for many types of cell lines. So again, this was done without any selection for successful transfection. Right. So this is again very different from what happens in bacterial cells, because this would never work in bacterial cells because so few of the cells would have taken it up. Right. So at this point, the nucleic acid that's being expressed, its plasmids, they're loose in the nucleus. They're not integrated into the chromosome, but there they will still be transcribed by RNA Pol II, thanks to the CMV promoter. So obviously, if we're seeing green cells, we know that the Pol II was working, the mRNAs were made, and the protein was being made, and exported as we wanted to see the protein of interest. All right, so we needed the E. coli stuff for this, the promoter, but not the ORI or the neogene. So we would need something like the neogene if we want to have maintained the plasmid in mammalian cells. So this is a process whereby some of the molecules actually become incorporated into the chromosome at some locations, or different locations really, and will therefore be replicated along with the chromosome in what are called stable cell lines. Okay, so very rarely the plasmid will actually integrate into the chromosome in the nucleus. And when this happens, the DNA will be replicated when the cell divides. So this is this concept of stable cell lines, in which the transgene is now part of the genome of the host cell. And these need to be selected because it's a relatively rare occurrence. Now, there's different ways of selection for this. I'm going to mention neo-resistant, well, it's really G418 resistance, because that's the most typically and straightforward in use. Okay, so this is genuinely confusing, I think. The neo gene, which is a bacterial gene actually, confers resistance to several drugs. It certainly confers resistance to neomycin and kanamycin in prokaryotes, but also this other drug called G418, which is most commonly used in mammalian cells. So G418 is a drug that inhibits protein synthesis when presented to normal cells. So this is your kind of same idea of combination of your antibiotic or anti-cell growth thing, and then a resistance gene. So in this case, the neo gene inactivates the G418 so that cells that have the neo gene can survive the presence of the G418. So it's the same idea of selection. It's just that the names of the gene and the drug are different because the neo actually inactivates several drugs. But the plasmid doesn't replicate until it's inserted into the chromosome. So ultimately, we have to use this sort of selection to acquire those cells that have actually maintained this plasmid permanently. And these are the stable cell lines. So the industrial uses need stable cell lines that express high levels of the protein of interest. And there's a lot of manipulation that goes into creating those. Okay, the plasmid shown below needs a CMV promoter for expression in E. coli cells, replication in E. coli cells, expression in mammalian cells, A and B, A, B, and C. Right, so the answer is C. The plasmid shown below needs an AMP gene for selection in mammalian cells, selection in bacterial cells, replication in bacterial cells, replication in mammalian cells. Right, B, selection. The plasmid shown below needs a GFP gene to select E. coli transformants, select mammalian cell transformants, observe successful transfection. Yes, the answer is C. And the reason is it's not a selection. We're just going to look at them. So we need the GFP that we can look at the cells and see whether they're green or not. So we're using it to observe rather than select for transfectants. Okay, lastly, there's other ways to show that you produce your protein of interest. One is to tag it with GFP as above, and the other is a method that we discussed a little bit earlier, known as epitope tagging. And this has the virtue that you're adding a few amino acids to a protein that correspond to a well-characterized and very specific antibody. So, of course, if you've already got a good antibody to your purified protein, you don't need to do this. But epitope tagging is a very common way to easily detect your protein of interest on a Western blot. Right. So the idea, of course, that would be involved in our initial choice of vector. If we think we're going to use an epitope tag, we want to choose a vector that, most commonly at least, contains the DNA sequences that encode those extra amino acids that you want that will be added to your protein by the process of transcription and then translation. So the vector itself contains the amino acids, and we need to put everything together properly in frame so that the extra amino acids end up stuck on the beginning or end of the protein eventually, even though what we're actually manipulating is the DNA codons. So the antibodies that are available are specific for these extra amino acids, and they will enable us to see our protein and sometimes to purify it, depending on what the tag actually is. Okay, epitope tags that are extra amino acids added to the N-terminus, C-terminus, either. The amino acids that are in the epitope tag are added to the protein, are encoded by the DNA sequence of the vector, are added to the protein after synthesis, are encoded by the mRNA that is produced from the vector, A and B, A, B, and C. No, A and B, A and C. So the answer is E, A, and C. They're encoded by the DNA sequence of the vector and are encoded by the mRNA that is produced from the vector. Yay.